So today we're talking about, this today should be different. Today we are talking about microscopes. So hopefully you all have your notes out and you can write down the title of the lesson as well as today's date and the fact that it is Unit 2, Day 4. But the objective for the day is biologists will be able to compare different types of cells from a virtual microscope lab and explain how to properly use a microscope and understand the major differences between cell types. All right. So this is kind of a review before we jump into the microscope jargon, a review of what we've discussed over the last 3 days. So at this point, you all know that all living things are made up of cells and that cells are the smallest unit of life, but we can further classify them as being prokaryotic or eukaryotic. So there are those two different cell types. Additionally, we can subclassify those two cell types even further and say that prokaryotic cells can be bacteria or they can be archaea, and eukaryotic cells can be plant cells, they could be animal cells, they could be fungi, and they can also be protists. And there are slight differences between these different cell types, but for the purposes of our biology class, we focus primarily on plant and animal cells and bacteria cells. But we use microscopes to be able to classify these cells, and this is really it gets into the history of science because we use these microscopes to really understand some of the functional and structural differences between these cell types. So microscopes and the advancement in microscope technology has really allowed us to better understand the differences between these cell types. And later in the school year, or in the semester, we will talk about a little bit more about that history, because it's important to know that at one point we simply classified things as plants and animals. We had no way of knowing that bacteria even existed. We didn't have microscopes. So if it was not visible to the naked eye, then we simply didn't know it existed. We had some guesses, but nothing scientific. And then once the microscope was developed, then we knew that we could classify things as plants, animals, and bacteria. But we started to recognize that they did not exist under the same domain, that there were major differences between them. And it's only been recently, in the last 60 or 70 years, that we've been able to introduce another domain, that being archaea. And so we'll talk more about that later in the semester. Okay, so in your notes, you have 4 minutes to get this Venn diagram down and try to fill it out with as many facts you know about animal and plant cells. So you all know how Venn diagrams work. And again, we're thinking about effective and efficient note taking. So this is a great example of a mind map note taking strategy. You want to get some differences between the animal cells and the plant cells. And then in the middle, you want to also have some similarities between them. So you've got 3.5 minutes. Make sure that you're working on that in your notes while I finish taking attendance. Okay, good. So hopefully you have some good stuff in your notes. Before we go on, I want to say a couple of things. I did thank you. I want to thank Alicia for pointing out some of the issues that she was having with the assignments yesterday, and they were totally not her fault. They were all my fault. And so I went in and I edited the assignments because some of them, similarly to the labeling cells assignment that we did on Friday, some of the assignments were not totally visible. The pictures didn't come up. So they're edited now. What you might need to do if you already started the assignment is submit it, but I've set it up so that you can submit multiple attempts. And so it'll save your highest score. But if you go in and you still are having issues accessing the pictures, you'll probably need to submit first and then try again. Okay, but they've been edited, so there's a Spanish translation there, and also the pictures are now going to be visible. Additionally, today is Jalen's birthday, so everyone wish Jalen a happy birthday. You can come off mute or you can send him a message.
Happy birthday, Jalen.
Appreciate you.
Jalen, how old are you today?
I'm 16.
Excellent, excellent. Well, congratulations. You know, I definitely am glad that I've gotten a chance to know you over the last year, starting with baseball, and now here you are in my class. So I know you to be a really strong student and a really respectful young man. I'm excited for you to take on this next year of life. Okay, excellent. So everybody's wishing Jalen a happy birthday, and we will move on. So today we are going to do a virtual lab. So I'll talk about that a little bit later, but if we have any issues with that on Canvas, then I definitely want you all to let me know. So you will probably have to refresh your Canvas page because I did just make a slight edit to it, but we'll talk about that in about 15 minutes. All right, so this is basically what you should have in your Venn diagram. Excuse me. And again, these Venn diagrams are excellent ways of note-taking. They're excellent methods of note-taking. For those of us who like to have things represented in a more visual and maybe even an artistic way, this is a great way to be able to organize your thoughts. We've got some differences, we've got some similarities, and you can even expand these Venn diagrams to include more than two categories as well. And that's when you really start to get creative with it. So this is something that you all should explore. Again, our focus is on taking the most efficient and effective notes. Efficient meaning we don't want to waste too much time. We want to be able to pull out the most important information and not have to rewrite everything that we're reading. And effective meaning that we really want to have the most bang for our buck. The time that we are spending, we want to make sure that the information is going to be useful to us, is going to be directly related to whatever our learning objective is for the day. So when we're thinking about the differentiation between cell types, this is essentially what you should have in your Venn diagram. I'll start with the similarities. Obviously, both of these cell types are cells, meaning they are living. They're both eukaryotic, and that's something that we showed on the last slide. They both have nuclei. They both have organelles. And that's essentially where the similarities end. Animal cells don't have chloroplasts because chloroplasts are required for photosynthesis. So animals do not need to undergo photosynthesis because we can eat food. We don't need to create our own food. Animal cells do not have a cell wall. The reason for that is that we don't need structure provided by water pressure. We have special cells in our bodies called bone cells or osteocytes, which give our bodies a structure. So we don't rely on cell walls. Typically, animal cells have a round shape that can vary a little bit, but we also know that they have a smaller vacuole because the need to store water is not as great. And then on the plant cell side, we basically have the opposite of everything that we just saw in the animal cell. But I'll just point out that, again, the chloroplasts and animal cells are required for photosynthesis. And they have a larger vacuole. Can someone just come off mute and tell me why, briefly, we require, or not we, but plant cells require that larger vacuole? I see you, Adrian. You're all good. Change my attendance now. I'll repeat the question. Can someone come off of mute and tell us why larger vacuoles are present in plant cells?
Because sometimes the energy that the plants need from the sun's light isn't always available.
Good, yeah. So we know that vacuoles are required for storing things, and so sometimes that light from the sun, for example, at nighttime, is not available to them, and so they need to have specific food stored in the vacuole. But what else do plants really need to make sure they're storing a lot of?
Water.
Yes, water. Plants have to store a lot of water. So that they have a much larger vacuole because they need to store water in case that they go through a period in which there is no access to water. So a period of drought or a period that is just a bit more dry. And so they store this water in order to remain a bit more rigid. And we'll talk more about that, what I mean by that in a couple of units. But if you've ever noticed, if you ever had a plant, when it hasn't been watered in a while, it starts to wilt and it starts to droop down a little bit. So that water is actually what's required to keep the plant in a rigid structure. Okay, now I'm going to push you all just a little bit with this question. So if we know that plants need to have large vacuoles to store water, specifically so that they can survive periods that are drier, what plants would we expect to have the largest vacuoles?
Cactuses.
Good. Yeah, we would expect that cacti have the largest vacuoles because they have to go much longer periods of time without water. And so any little bit of water that they can get, whether it's from the soil, whether it's from a reservoir that's deep underground, whether it's from animal waste, whatever amount of water they can get, they need to be able to store that for a long period of time. So they have large vacuoles that are very, very efficient at storing water. Thank you, Gabby. All right. So today we're talking about microscopes, and this is going to be a quick lesson. We only have about 12 minutes left in the synchronous portion of class. But microscopes are extremely useful, not just in biology, but in essentially all fields of science. And you all are probably pretty familiar with this very basic microscope. It's got an ocular lens and an objective lens. So the ocular lens, ocular is a word meaning eyes. So this is actually where you would place your eyes to see whatever is being objectified. And then the objective lens is also required for magnification, and it's what's directed at the object. So you would place your slide right here, and the objective lens would be directed at that slide. There are some other important pieces of the microscope that we would point out if we were in a physical lab, but you all will get a chance to see them during our asynchronous portion of class today. So I won't talk too briefly about them, but this small technology is a result of many, many years of hard scientific work. And it's really, really quite fascinating that we have this technology that allows us to magnify things 500 times their normal size or their actual size. So you all will see in the virtual lab, but hopefully when we are back in school, you'll get a chance to see some of the slides that I have in the classroom. And you'll be able to see what a muscle cell looks like, what the cell from a lily pad looks like, what an eye cell looks like. And I think it's really just an important part in growing your fascination of science. Okay, but each microscope and each objective lens has a different amount of magnification. And so they can enhance things to different sizes. And the total power magnification is how we measure the amount of enhancement that can take place. And it's just a very easy equation. We take the power of the objective lens and we multiply it by the power of the ocular lens. Oh gosh, it's being slow. Okay, so the ocular lens almost always has a power of 10 unless you're dealing with a much stronger and more expensive microscope. So we can almost always have this red number as 10, and then we just multiply the objective lens by 10 to get the total power magnification. So practice question. We're looking at a blood cell or a slide of blood cells using the 10 times objective lens. So what would be the total power magnification? And we're just going to assume that the ocular lens is 10 as well. Based on this equation right here, very simple equation, take the power of the objective lens times the power of the ocular lens. And the ocular lens almost always has a power of 10. So in this case, the objective lens also has a power of 10. So what would be the TPM?
100.
Thank you. It's just 100. We just do 10 times 10. So pretty simple there. All right, so this is what your blood cells, red blood cells, would look like at a magnification of 100 times. Objective lenses can rotate, and so we can have magnifications of 4x, 10x, 40x, or 100x. So a magnification of under an objective lens of 40x, the total power of magnification would be 400 times. So this is what your hair would look like, or not your hair, but a strand of hair would look like under a TPM of 400. And then this is what it would look like under a TPM of 1000. And here's 4000. So we can see some clear differences And we talked about this briefly because Ayana asked the question. But there are two different types of microscopes, essentially. Though there are many more types than just two, but two that we are familiar with based on the pictures that we often see. A light microscope is cheaper and it provides minimal detail, whereas an electron microscope is expensive and provides a great deal of detail. The purpose of both, essentially, and we can use this in forensic science if we're trying to identify specific genetic markers. We can use this in evolutionary science if we're hoping to identify a species or see similarities between species. These microscopes allow us to observe a variety of cells and then to determine differences between those cell types. So this is what I was saying when I talked about the fact that microscopes are important for every field of science. Even in certain fields of chemistry, we use very powerful microscopes that don't allow us to visualize atoms, but they can give us specific character traits of atoms. So I can know that a substance is made up of 10% carbon and 40% hydrogen and 20% oxygen, so on and so forth. So microscopes give us a much greater idea of what's happening on a scale that we would not even know existed if these microscopes didn't exist. Okay, so here's an example of the difference in strength of the two. So your light microscopic image is on the left, and your electron microscopic image is on the right. The way an electron microscope works is that it will shoot electrons at your object, and the electrons are bounced back in a specific pattern. And based on the size and the density of the object, the electrons get bounced back and they can create an image of the object. So the image on the right of the ant right here has been colorized, which is not actually how it would appear, but you could get it in such a great detail. Here are more examples. Here are your light microscopes. Here are red blood cells under a light microscope. Here they are under an electron microscope. I have no idea what this prehistoric dystopian insect is. It could be a mite.
What's a mite?
Like a bed bug, essentially.
Yeah.
And then these are spores from plant cells. And here's a picture of plant cells under a light microscope. So clear and very obvious differences between the two. And then you can see that there are big differences in size between the two as well. An electron microscope involves a desktop computer as well as a large desk and this very large apparatus. So extremely expensive, difficult to operate, takes a lot of training, versus a light microscope, anybody can do it essentially. And if we were in class, in school, you all would be doing it. All you have to do is look into it essentially and turn the light on. All right. This slide there. Okay. So at this point, what I'll have you all do is go into Canvas, and you'll see that our asynchronous assignment for the day is called the Virtual Microscopy Lab. U2D4. I'm going to ask you to make a copy. Once you click on the link, you'll have to make a copy, save it in your biology folder, and that's where you can work on it. Please let me know if anything does not work.
What do you mean biology folder?
Your biology Google folder.
Um,
The one that you made in the first week of school, and you saved it in your Google Drive.
no, I don't remember.
Really?
I don't remember.
Okay, I'll send you a link for yours in 10 seconds.
Okay.
Maddie. No. Oh, yeah. There's like new materials that you can like.
Oh, apparently, Amari, I don't see yours. Maybe you never made one. Let me see. Yeah, I don't see yours. So you need to make your Google folder.
What am I supposed to make it on?
If you go into Canvas, there's an assignment in the Unit 0 module.
Unit 0?
Yes. There's an assignment there called Create Your Google Drive.
Which day? Oh, great.
And Alicia, yeah, it's for your question, Amari, it should be under day 2. And then Alicia, for your question, it should be, it looks published.
No, please.
You may need to refresh your Canvas page, but it should be under day 4.
You felt that, or did you see that? I saw it. I cried every time I saw it. Dang it. Chris, I don't leave like that. I made it. Oh, no, I used it. I felt. My dog is looking at my arm like it's a chicken bone. No. You may just like skip this level. It's super hard. No, they're using Udo down there. Oh my goodness, who is that?
Mr. Rod.
Right, I just got it.
Okay.
So now you can save this new assignment in that folder.
Mr. Rudd.
Yes.
I don't see that assignment, but I just done the exit ticket.
You probably will need to refresh your page because I edited it, so it probably may have disappeared for a minute. But just refresh your page and then check again. Let me know if you still don't see it.
Mr.
Yes, sir.
How are we supposed to put the virtual thing inside of our biology folder?
Can you share your screen? That way I
Yeah.
can show other people who might have the same problem.
All right, I'm finishing my screen. All right, I started sharing it.
Okay, here it comes. All right, go back to your assignment. The Google Doc.
Oh.
First of all, make sure you change the name. So just put just put Amari Sanders Richardson, virtual lab. Good. Okay. Now, if you click on File, you see it says Move.
Right here.
Yep.
Click on it.
Yep. Okay. So now, now you just click on that arrow first. Okay. Hold on. Okay, that's good. Yeah. Click Move. And all right, let me make sure I have access to it. Yep, it's there.
Okay.
Thank you. Jalen, it should be, make sure you're looking in the Unit 2 module under day 4. Jalen, stay on for a second so you can share your screen. But everybody else, it is 11:35, so you are good to go. Thank you for being here. I'm going to stop the recording now. And I will talk to you all tomorrow. We're going to just review before our quiz on Friday.